In this video, we're going to start our discussion of DNA. So the title of this section is Using Microbiology to Discover the Secrets of Life. And the focus is going to be on really the discovery of DNA. So back when we had the establishment of the germ theory of disease, we didn't know anything about DNA. We didn't know anything about how heredity worked, really. We didn't know about how traits were passed, from one person to another. We had no idea about the heredity of bacteria. So there was this misconception that heredity involved a blending of traits, that the offspring would get some characteristics from mom, some characteristics from dad, but that there would be a blend. So, for instance, if your dad was really tall and your mom was really short, then you'd be somewhere in the middle. Or if, your dad had black hair and your mom had blonde hair, well, then you would have brown hair. So they believe that there was this blending of traits. So in the mid to late 1800s, we started to really research this and try to discover really what was in control of heredity and therefore really starting the field of genetics. So the first person who kind of came on board here was Friedrich Miescher. And this was in the 1860s, so about 150 years ago. He was a doctor, and he actually isolated some chemicals from white blood cells that he got from pus that was on the bandage of a wound. He named them nuclein at the time because they came from the nucleus, but he didn't really know what they were. And then a few years later, his student, Richard Altman, named it nucleic acid because he discovered that it had acidic properties. And then we had more work on this by Cosell, who actually isolated the different nucleotides that are found in the nucleic acid: adenine, guanine, thymine, cytosine, and uracil, which we'll get to in a little bit. And so he actually received the Nobel Prize for his work with this. So we had people working on DNA. Didn't know that it was DNA at the time, but this is kind of the precursor research that led to us understanding what DNA was, and then trying to discover the structure of DNA and how it actually contributed to heredity. So we had this discovery, but we didn't really know then the importance of that. So then, also in the 1850s and 1860s, we had Gregor Mendel, who was an Austrian monk, and he was in charge of the garden, or he worked in the garden of the monastery. And one of the things that he became interested in is inheritance. He was using peas. So he's working in the garden. These peas were readily available to him. So he started to research genetics, basically, the heredity patterns. And so he used these pea plants because they self-fertilize and it's highly inbred. So you get what are referred to as true breeding pea plant lines, which are plants that always produce plants that look identical to them. So he was able to study 7 different traits and tracing them through 2 generations, this F1 generation and F2 generation. And so what he found is he took a hybridization. He said, he had a plant with violet flowers or purple flowers and a plant with white flowers. And again, these were true breeding. And he said, I wonder what's going to happen if I cross them. And so, based on the knowledge of the day, we would expect to get a lavender color. We would expect to get a light purple, a mix between the violet and the white. Or we would get, some purple flowers or some white flowers. But what he discovered is in the F1 generation, that first generation, all of the flowers were violet. They were all purple. It was like the white had just disappeared. So then he allowed self-fertilization here of this plant, and what he found is that in that offspring, in that generation, that what we call the F2 generation, that we got about 3/4 of them would be purple flowered plants, and about 1/4 would be white. So he kind of got this 3-to-1 ratio that appeared in this F2 generation. And he experimented, you see there, with nearly 30,000 pea plants. He presented this in 1865. It was published in 1866. And it was not really accepted at the time. It wasn't until we discovered DNA, really, that his work was kind of rediscovered and it started to make a lot more sense. So we consider Mendel the father of genetics because his work really laid the foundation for understanding how inheritance patterns work. Now, because he was carrying out his experiments before we knew what a chromosome was, this was kind of part of why people didn't really understand or really believe in his work, because we didn't know anything about chromosomes. During the late 1800s, going into the early 1900s, we actually were able to stain these nuclear structures and watch as cells divided using the microscope, because we had developed more powerful microscopes. So in 1902, an individual by the name of Boveri observed these chromosomes, what we came to call chromosomes, in sea urchins. And that same year, another individual, Sutton, observed the separation of the chromosomes into daughter cells during meiosis. So he was watching cells that were going through these processes of mitosis and meiosis, and he was able to see how they were transmitted, or how they got from the parent cell into the daughter cell. And this led to the development of what we now call the chromosomal theory of inheritance, and that is what we now know is carrying the genetic material that was responsible for these inheritance patterns that Mendel observed. And then in the 1910s, around 1915, we had an individual named Thomas Hunt Morgan. He worked with the fruit fly Drosophila melanogaster. And ever since Morgan started his work with fruit flies, these have really been the organisms that have been used extensively in genetic trials, because they grow so quickly and they're easy to observe and they're easy to, keep in a closed area so that you don't get any crossbreeding or anything like that. And so Thomas Hunt Morgan published The Mechanism of Mendelian Heredity, and he received the Nobel Prize in 1933. So he basically followed up on all of Mendel's research, proving Mendel right. Mendel just didn't know about chromosomes because chromosomes had not been discovered when he was doing all his work. So really, with the work of Boveri and Sutton, Thomas Hunt Morgan was able to put it all together with Mendel's work. And then in the 1920s and then on into the mid 1900s, we had Barbara McClintock, who developed different staining techniques that allowed her to visualize different chromosomes. And she worked with corn. And so she identified these, what we refer to as transposons. Transposons are known as jumping genes. They are genes that can actually change places on a chromosome. And so those are called transposons, and we'll talk about those a little bit later on, but they can regulate gene expression, what genes are turned on and off. They can regulate protein expression, so what proteins are made. And when we're looking at bacteria or even viruses, the ability to cause disease. So in looking at this work that was done by all of these individuals, we now have an idea of how all living organisms work in terms of their chromosomal inheritance. So there's a saying here in your book: what is true for E. coli is also true for the element, meaning that the same biochemicals that allow E. coli to do what it does are found in the elephant and allows the elephant to do what it does. So we're all made up of the same, basic building blocks, the same basic components. It's just the complexity and what we can do with those that make us all different. Now, once we had this idea of a basic biochemical entity that was responsible for inheritance, a lot of other scientists performed a lot of experiments on a lot of different organisms. We were really trying to understand, okay, so what's in these chromosomes? What's going on there? So another team of scientists, BIOL and Tatum, they worked on a particular bread mold, and they were trying to determine the relationship between genes and proteins. So we have this idea that we have these chromosomes, we have these genes on them. Then how do we get to the proteins, essentially? And so BIOL had worked with Morgan in the fruit flies and found they're a little too complicated, so let me pick something simpler. And so they chose mold. And what they did is they took the molds and exposed them to X-rays. And X-rays have the ability to cause mutations, changes in the nucleic acids. And what they did is they took those irradiated spores, mated them, and attempted to grow them. And they looked for mutants that could grow on this complete medium. The complete medium would have the vitamins and amino acids that they needed, but these irradiated ones couldn't grow on the minimal medium because it didn't contain the nutrients that they needed. And so those molds then would contain mutations in the genes that encoded for those biosynthetic pathways. So then they found these mutants and then they tested them. Okay. And so then they're tested on a medium that contained just a particular type of amino acid. And so spores that grow only in that one particular amino acid would have a mutation in that pathway that produces that amino acid. So what they said then, or what it led to, is this one gene, one enzyme, or one gene, one protein. So this hypothesis basically said that we have these chromosomes that have these different genes on them. And each one of these genes is going to code for a particular protein. So this led to the one gene, one protein hypothesis, that, all of these different genes that we have are going to code for all of these different proteins. And genes then are basically the code for all of these different proteins. And these proteins are what make living organisms what they are. So although there are some genes we know now that don't really code for proteins, they code for other things. For the most part, this one gene, one protein hypothesis is true. And so that was BIOL Tatum's work, and that led to their 1958 Nobel Prize win.